Hello, everyone. Thank you for joining. Good morning, good afternoon, and good evening to everyone. I am Dr. Mahalakshmi Arujanan. You can call me Maha for short. I'm the Global Coordinator of the International Service for the Acquisition of Agri-Biotech Applications, or in short, ISAAA. ISAAA is today very proud to join Outreach Network for Gene Drive Research with ISAAA's Biotechnology Information Centers spread across Asia. And we are extremely happy to contribute towards greater understanding of gene drive research through a 4-part webinar series. This is the first one. So with us today, we have got over 100 participants. We initially got registration from 48 countries, showing the great interest the stakeholders are showing in this new technology. We all know genetic modification and gene editing are common gene technologies. We also have gene drive, which is another genetic approach that potentially offers direct benefits to biodiversity conservation, public health, and the control of pests that harms human, agriculture, and biodiversity. Given its complexity and novelty, we feel the need to create more platforms to discuss gene drive research, its potential and responsible use, as well as reflect the current international discussion surrounding this technology, such as under the Convention on Biological Diversity. So in this 4-part webinar series, we will be discussing the basics of gene drive, which we will be doing today, its role in protecting biodiversity and eliminating vector-borne diseases, and finally, gene drive regulations and governance. Before we start the webinar, let me just briefly provide some housekeeping rules. We have 2 speakers today, and the discussion will be at the end of both presentations. You may post your questions, suggestions, comments throughout the webinar on the Q&A feature. The webinar is also live streamed on ISAAA Facebook and YouTube channels. So you may also, if you're watching it from those channels, you may also post your comments and questions there. We will be picking up your questions from there too. With that, I will now introduce our moderator for today, Ms. Crystal Birunji Mirsiga. She's a field entomology coordinator at Target Malaria Uganda, based at the Ugandan Virus Research Institute. Crystal holds a double major in zoology and botany from Makerere University. And Target Malaria Uganda is part of the Target Malaria Consortium, which is a not-for-profit research consortium supported by the Bill and Melinda Gates Foundation and the National Institutes for Health. Target Malaria aims to reduce the malaria transmission in sub-Saharan Africa by reducing the population of malaria-transmitting mosquitoes. Crystal herself is involved in research on Anopheles gambia, focusing on population dynamics, behavior, and species composition across Uganda. So with that, I'm going to invite Crystal to come on screen to moderate the webinar. Welcome, Crystal.
All right. Thank you all for joining us today. Please allow me to introduce our distinguished speakers for this event. First, we have Professor Paul Thomas. Professor Thomas leads the South Australian Genome Editing Facility at the University of Adelaide and the Genome Editing Program at the South Australian Health and Medical Research Institutes. He works on a wide range of applications of gene editing technology, including the development of gene drives to control invasive species and the development of genetic therapies for inherited diseases. Welcome, Professor Thomas. We also have a second speaker, Dr. Prasad Pradkar. He leads the Vectorborne Diseases group at the Commonwealth Scientific and Industrial Research Organization. He has extensive research experience in researching vector-borne diseases, including experiments with genetically modified mosquitoes. You're very welcome, Dr. Pradkar. So let's begin with Professor Paul Thomas.
Many thanks, Crystal, and the organisers for the opportunity to tell you a little bit about gene drives, a little bit about how they work, and I'll just touch on also some of our research as I go through the presentation today. So let's get started. So I'd like to set the scene and give you some really some insights into how this new technology works and how it's being used in a couple of different instances. And I guess the first thing I'd like to say is that we talk about gene drives as though it's a single concept, a single type of genetic modification, and that's not really the case. There are many different types of gene drives that we can think about and design. And indeed, there are many gene drives that are naturally occurring genetic modifications that occur in nature, and we've known about these for a long time. I guess the excitement around gene drives is really around synthetic gene drives, and in particular, the emergence of this new Crispr technology, which I'm sure will be familiar to many of you, has really, really given a lot of impetus for this research. And as you'll see, it's really one of the driving forces behind this new wave of technologies that are emerging. So again, just so we're all on the same page, this will be familiar territory, I'm sure, to many of you in the audience, but just to, I guess, start with genetics 101, as it were, just to remind you that in every cell of every organism, really, there are the DNA building blocks of life. And in the case of mouse, I'm just using it here as an example, we have 20 chromosomes, and they contain altogether around 20,000 genes. And these genes are distributed across the chromosomes, and they perform different functions. So each gene has its role, and if we damage the gene by changing the sequence of the 4 DNA bases, we can alter its function, often inactivate it, so that the organism develops some kind of phenotype, for example, infertility, associated with damage in a particular gene, and that's called a knockout. I'll talk a little bit about this concept called transgenesis, and that's really just a term that's used to describe adding a foreign piece of DNA into the genome. And this is used in many contexts, and this can add a new phenotype or a new capability to the organism by virtue of the fact that it's got a new gene to do something with. And all gene drives are, at least in the context of what we'll talk about today, are transgenes that have a very special function, and they're able to either replicate themselves or to influence sperm or oocytes in such a way that they get passed on to the next generation with greater than 50% frequency. And that's what a gene drive is. So let me explain that in a little bit more detail. So when we think about trying to modify a population, if we just made a normal transgenic organism, so in other words, we added an extra gene into the, for example, let's say we put in an extra gene on both copies of chromosome 15, what would happen would be, under those conditions, would be that transgene would get passed on to 50% of the progeny once it was, if you like, heterozygote, in other words, present on one of the chromosomes, but not on the second chromosome. And that's not very useful for modifying a population, because it will simply be diluted out over time. So what makes a gene drive is this ability for it to, as I said, either replicate itself or somehow change the way it's passed on to the next generation, such that it's always inherited, or at least inherited with greater than 50% probability, which is obviously the normal inheritance pattern. So here it is in schematic form, if you like, in terms of a pedigree or a population. We can think about a gene drive as something that will spread through the population because it's always passed on to the offspring from a parent. And as you can see, this can actually quite rapidly, if it's passed on at 100% of the time, this could very rapidly spread through a population. So it allows population level engineering, which is very useful for many contexts. I'll tell you about a couple in a moment. And it can be used to modify that population. For example, you can imagine putting through a malarial resistance gene in the case of mosquitoes, or you could use it potentially to suppress the population by, for example, spraying an infertility gene. And I'm going to give you some examples of that as we go through the presentation. Okay, so why do we want to develop gene drives? And there's lots of discussion around this. And of course, many people are excited about population level modification, because it enables us to do things that we really haven't been able to do before. And one of the key examples, of course, is malaria, where this research is most advanced, and we'll hear more about that in the following seminar. So I'm not going to talk really too much about that today, except to touch on a couple of examples to make points. Where our research is heading is for a different application, and more around the use of gene drives for conservation or even agricultural benefits. So particularly on islands, when rodents get onto an island as a feral pest, that is either mice or rats, they cause enormous damage because they can, for example, eat the chicks or the eggs of ground nesting birds. And it's quite clear that in many examples that rodents have a devastating impact on biodiversity on islands and are responsible for many species losses in that context. So there is a really good reason to develop new technologies in that context to help us with this long-term issue of rodent damage on islands. And gene drivers is one of the technologies that's being considered for that. The other one is particularly close to us here in Australia. We have regular mouse plagues, and at the moment we are in the middle of one of the worst mouse plagues we have seen in this country. And again, there's lots of call for new technology to help us manage rodent populations at scale. And again, gene drivers being considered and in fact developed for this purpose as well. Of course, there are many aspects to its deployment. We'll talk about them at the end. So how would you generate a gene drive? And this is a question I get asked quite often. And it's not, this is not something that we would do, within a few weeks or even within a few months. Typically, these kinds of experiments take years to perform. It depends, of course, on the model species that we're talking about. I'll give you a perspective from mice because that's the species that we work on, but the same principles apply regardless of which type of species you want to develop a gene drive in. First of all, you need to think about what strategies you want for your gene drive, and there are two that I'll present to you today, but there are many, many to consider. You need to generate some DNA constructs to test in the laboratory. These are effectively the components of the gene drive. Often you'll test those components in a cell culture system before going to the bigger step, or if you like, more sophisticated step of actually generating the animal itself that contains the gene drive construct. You have to then validate those new animals, and then finally you get to do the experiment. And this is just the initial experiment, a so-called cage trial. Again, I'm showing you mice here, but this would also apply, for example, in mosquitoes or flies or whatever system you're working with. So this takes a really, 1 to 2 years to develop all this experimental tools and actually do the experiment. And if your experiment works, then you can think about the steps that could take you towards deployment. And there are many. And we're not going to talk about social engagement today. We're not going to talk about safety mechanisms for gene drives. We're not going to talk about regulation specifically, but I just want to make the point that all these things, of course, need to be considered on the pathway towards deployment of a gene drive technology. And there are many things that go on in the background that you need. Obviously, funding, you need staff to do these things. You need to engage the regulator. In Australia, we have quite a strict regulation around even experimental development of gene drives and, of course, ethics as well. So the two strategies I'm going to tell you about today are ones that are, I guess, more advanced and discussed probably in the context of more species, and probably would be the ones that are going to arrive first in terms of potential deployment. But again, just to make the point, there are many different types of gene drives that people are developing in a laboratory setting as proof of concept, and they may also in time reach maturity in terms of active deployment. Just one slide on this. Again, I imagine many of you will be familiar with CRISPR-Cas9 gene editing, but for those of you who are not so familiar with this technology, essentially this is a pair of molecular scissors that we can design to cut virtually any sequence within the genome. It's a two-part system. The Cas9 is the green blob here, which is a protein that does the cutting, and it's guided to its address by this blue line, the so-called guide RNA, and this base pairs with the target sequence to generate the double-stranded break at the site of interest. And suffice it to say that this technology has enabled us in many ways to progress gene drive, synthetic gene drive research by enabling us to generate the transgenic organisms themselves much more quickly. It's involved directly in this process of homing, which I'll tell you about as one of the gene drive strategies. And it's also involved in the chromosome shredding, which I'll tell you about as the second gene drive strategy. Okay, so this first strategy is the one that's the most advanced, and it was the one that was published as the first synthetic gene drive a number of years ago and got people really excited. I'll show you the publications in a minute, just so you have something to go away and read if you want to get more information about the history of this research that over the last few years. But essentially the idea is as follows. This could be any organism, but we're, as I said, trying to develop it in mice. So you have a gene drive, which is the Cas9, that endonuclease, the protein that makes the cut. You've got its guiding molecule here, which enables it to cut at the right sequence. And there's an optional cargo that you might want to add on to the gene drive as well. And this is a transgene that is integrated into the chromosome of interest. So here's our gene drive mouse. It's got 2 copies of the gene drive, 1 on each chromosome. And just to show you how it works, we'll have a simulator mating with just a wild type mouse, so a mouse without the gene drive. So what happens in the germline of the resulting mouse, it's got 1 copy of the gene drive shown here and 1 copy of the wild type chromosome. The gene drive is engineered in such a way that it makes a break, a double-stranded break in this target, this wild type chromosome, at exactly the point where the gene drive itself is integrated on the other chromosome, the homologous chromosome. And the germ cell needs to repair this double-stranded break, otherwise it will not survive. So what tends to happen, if the experiment goes well, is that it uses the gene drive sequence chromosome, this chromosome, as the repair template to repair this double-stranded break. And as a result, you get now 2 copies of the gene drive, 1 on each chromosome, the original one and a second copy, which is made as part of this repair process. And that process is called homing. And the reason it's important is that we now have copies of the gene drive on both chromosomes, which means that it must therefore be passed on to the next generation. So quite a clever little strategy that was worked out a number of years ago. Now, it doesn't, of course, this is biology we're talking about here, it doesn't always go according to plan. And what we've also realized in recent years is that an alternative repair process can occur. I won't get into the details, but it's called non-homologous end joining, or just sometimes end joining. And under this, when this occurs, instead of the homing process, you instead get just a little break. You get repair of the double-stranded break, but it loses a bit of DNA typically as part of that repair. And unfortunately, that means that it can no longer be susceptible to the gene drive because the sequence has changed at the target site of the CRISPR-Cas9 complex. So they're called resistance alleles, and they are, if you like, the enemy of gene drive spread. They're what these resistance alleles are going to be what will stop the gene drive spreading through the population. So as I said, there was a lot of excitement around a paper that came out in 2015 from Gantz and Beer at University of California, San Diego, showing that in Drosophila, so fruit fly, a well-known genetic model organism, what they called the mutagenic chain reaction, which is exactly the process that I've just described to you, a homing gene drive that they made synthetically using CRISPR-Cas9. And I guess even more excitingly, their group with Collaborators and other groups as well in London showed that you could use this technology also in Anopheles mosquitoes, which is important because we're talking now about a malaria vector. So this is now becoming on the radar as a technology that could potentially be deployed in the context of malaria. And what's astounding really is that in these Anopheles mosquitoes, the homing rate, so in other words, the good reaction that you hope to occur, is occurring in up to 99% of the time. So that's a very high bias towards transmission of the gene drive through the population. So really quite amazing ability to spread through a population. And indeed, just to give you a feel for, we'll probably hear more about this in the next from the next speaker, but just to give you a feel for a recent paper on this, I thought I would, just to show you the potential, how far advanced this has come even in the last few years. A recent paper showed that in a cage trial, so this is a reasonably large cage trial in mosquitoes, that you could crash the population within 8 or 12 generations, depending on 2 different experiments, using this gene drive approach. And in this particular example, it created infertile female mosquitoes. And there's lots of, there's a large risk assessment that's now going on and discussions going on underway towards the possibility of deployment, although that's still, I imagine, quite some time away. All right, so in terms of, you might be wondering, okay, that's great for mosquitoes. I started talking to you about conservation and in an island context with rodents. Can you use this technology in other organisms? And there are some experiments on this, and essentially what our group is trying to do, and there are a couple of others as well we know of, is to try and create a form of a gene drive which is exactly the same as I've been describing to you, that's come out of Drosophila and now mosquitoes, to control rodents on islands. And this is the kind of approach that we would like to develop. Again, we've got a gene drive, and this time it's positioned within a gene that's required for female fertility. In the germline, we would look to cut, as I've described to you before, and using that homing reaction, generate 2 copies of the gene drive, which would mean that we'd now always pass on that gene drive to the next generation. And in this phase, it would spread through the population. So as gene drive bearing individuals met with wild type individuals, all of their offspring would carry the gene drive to the point where it would eventually saturate the population. And at that point, the gene drive bearing individuals would start to mate with each other, and they would result in infertile offspring, because the gene drive would be knocking out the female fertility gene on both chromosomes. So this is the kind of approach we're looking to develop in mice. And just to give you a feel for how these experiments are going, it's very early days, and we have published a paper, and there's also a paper that's come out of UCSD, actually again in collaboration with the original Drosophila gene drive researchers, Gans and Beer. And the take-home message here is that it's not working anywhere near as well in mammals, in this case mice, as it has been working in the insect species. So I'm not convinced that this approach is going to work in mice. We need to essentially do more work to understand more fully whether it's going to be possible or not. So I guess it's a watch this space in terms of future research along this line. But I think it also underlines that different species are probably going to have different susceptibility to that gene drive mechanism that I've described to you. So we need to think about other strategies as well. And for the last couple of minutes, I will tell you about a second strategy that again has been partially developed in insects. So this strategy you also may have heard of. It's called the X shredder, or also sometimes known as a driving Y. And the idea here is that you can convert the population or deplete the population of females so that only male offspring are born, and that will cause the population to crash over time because there are no females to sustain the population. The way that it works does not require this homing mechanism that I've described to you. Instead, you look to again use CRISPR to shred the X chromosome. So during sperm development in males, you eliminate the X chromosome bearing sperm by actually degrading the whole X chromosome. And that's done by targeting repeat sequences that are specific to the X using the CRISPR machinery, which is present on the Y chromosome. So if that all goes according to plan, what happens is that you only generate sperm with a Y chromosome, and that Y chromosome contains the CRISPR gene drive, which means that in the next generation, from any cross between a wild type female and a gene drive male, you only generate males, and they all contain the gene drive. So in other words, it'll spread and it'll only generate males. So to give you a little bit of a feel how this could work, again, in a rodent setting, we've done some modeling, which I wanted to share with you. This is not published yet, but just to give you a feel for the potential of the technology. And if I can get it to start. Yes, here we go. What you're looking at here is an island, a virtual island. So this is all computer modeling and based on individual-based modeling. And we're deploying into this 200,000 population island the gene drives, which you can see here in red. And as they spread through the population, generating males, eventually we see population collapse. There's a little bit of emergence here at the end, but ultimately within around 4 to 5 years, we've managed to eradicate the island, at least a virtual island, using this driving Y or X shredding approach. So we think it does have a lot of potential if we can get it working in mice, and we're actually close to getting some data on our first experiments now. As I said, it has been developed to some extent in mosquitoes. So really, this is just to point out that there's not a new concept, at least in terms of being developed in rodents. There are some precedents in mosquitoes indicating that it could work. And those papers and this group has been working in this space for quite some time. And I think this also has promise for malaria. Perhaps we'll hear about that also in the next talk. So I hope altogether that's given you a little bit of a feel for where the field is at in terms of gene drive development. It's still relatively early days, and there are lots of issues that we need to address with this quite new and powerful, exciting technology. Essentially, in terms of take-home messages, you can think of gene drives as a genetic tool that can be used for population modification or suppression. And it's the fact that you can affect entire populations at landscape scale that gives it its enormous potential, either for human health benefits potentially, or conservation benefits, or even agricultural benefits. Gene drive technology is most advanced in the insect world, being driven largely by its potential for malaria control. But I hope that the work that we're doing and other similar groups are going to catch up in terms of its potential application in mammals as well. Happy to talk about that. And lastly, I think it's really important to understand that gene drive technology is not enough in and of itself to have been developed. We need many other pieces of the puzzles to come together before it could truly be deployed in a way that would benefit society. And that's going to involve, of course, public discussion about the technology, social engagement, that is, extensive modelling, regulatory approval, and even the incorporation to some degree of safety switches within the technology itself that would limit its impact to the target population of interest. All right, with that, I'd just like to mention that I'm part of a consortium called GBIRD, Genetic Biocontrol of Invasive Rodents, that is looking at all aspects of gene drive development that I've just been mentioning in the context of rodents and mice to start with. And lastly, just to acknowledge the wonderful workers in my group who actually do the work in this space. And some of them are mentioned here. Gelshan Chandran, who's now working with another group in Macquarie in Sydney, Maciek Maselko, Luke, who's working here as a PhD student, and Mark as well as a postdoc, and Sandy Piltz for generating all of the animals. So with that, I acknowledge my collaborators, my funders, and would like to leave it there, and happy to take questions once we get through to the end of the next presentation. So thank you very much.
Thank you, Professor Thomas, for that wonderful presentation. It was a very clear explanation of what gene drives are and how they function, and quite insightful as to the science behind them. At this point, I would like to turn over to Dr. Paradka to help us situate gene drives with regards to the history of their development and relationship with the comparable technologies. He will also point to precedents and experiences that we can learn from. Dr. Paradka?
Thanks, Crystal. Yes, thank you, Crystal, for that introduction. And of course, thanks to ISAAA for giving me the opportunity to present here. Hope you can see the screen. Yep. Okay. Yeah, as was mentioned before, my expertise is really in vector-borne diseases, so mosquito-transmitted diseases. And I'm going to talk about gene drive in sort of that context mostly, but obviously a lot of the information can be extrapolated to other systems, as Professor Paul mentioned before. My idea is to also give some context as to what we can learn from previous experiences using novel technologies. So I'll start with vector-borne diseases and why we study them. So vector-borne diseases are diseases transmitted by insects like mosquitoes, chilocoides, tsetse flies, as well as things like ticks. And there has been increase in this number of these diseases over last decades. And that's mainly because of three things. One is global climate change. So as the climate is warming, the geographic distribution of these mosquitoes and insects is increasing, leading to more transmission. There's also urbanization, and by urbanization, I really mean rapid urbanization that's happening all around the world in many cities. And of course, travel and trade. So, an infected person from one region can be in another region within a matter of hours. So the important thing is there is no really specific therapeutics available against any of these diseases. And very few of these diseases have any effective vaccines available against them. So it produces a really high burden on public health worldwide, and it's really focused in this tropical and subtropical regions. So the focus for public health is really on vector control, how we can reduce the population of vectors like mosquitoes in this case. Now there is a long history of vector control, starting from the end of 19th century with Sir Ronald Ross, with identification that malaria is really transmitted by mosquitoes, Anopheles mosquitoes, as was mentioned before, but also others who identified other diseases transmitted by mosquitoes, and that if we can reduce the population of mosquitoes, we can reduce the transmission. So that really put the pressure on public health to reduce the mosquito population. And initially the focus was on environmental control, and that was really removing stagnant water. As you probably all of you know, mosquitoes breed in water. So if you remove that water source, you can reduce the mosquito population. It's not always possible to remove all water sources, and that's why there is still mosquitoes breeding in various sites. There was also some advances in chemistry, which identified some insecticides initially. And these were non-persistent insecticides like pyrethrum. And by non-persistent, it means that these insecticides, once they are sprayed, they don't stay on that, stay in the environment for very long times. So it's in matter of hours or day, they are just removed. In late 1930s, scientists identified a chemical called DDT, and it was really a game changer because it was really effective. And it was one of the persistent insecticides, which means it stayed on in the environment for days and weeks. So it was heavily used in 1940s and 50s to reduce the mosquito population, and it was very effective. So WHO at that time declared that we need to have a global malaria eradication program, and we can achieve that with the use of DDT. However, it's in 1962, a book was published, which really changed public perception. This was a book called Silent Spring by Rachel Carson. And it really pointed out some inadvertent and long-term effects of DDT usage on other insects and other environmental, like birds and other animals. So with this led to a ban on DDT. And that was mainly because of pressure from public. And because DDT was banned, it was not used, and the mosquito population really rebounded, and it came back with resistance towards DDT. Now, since then, we have been really playing catch up with new insecticides and mosquito becoming resistant to those new insecticides. So there is a need for this novel technologies to be used. And some of the technologies that were developed in the middle of last century and in 50s and 60s, one of the technologies worth mentioning is the sterile insect technology. So this is where you can take a large number of mosquitoes and sterilize them using X-ray and release them in the wilds. And by this X-ray sterilization, this technology was initially used in 1960s by North America, by US. as for the screwworm sterilization program. And it was very effective. It basically wiped out the screwworm population in North America and was very successful. So this technology has still been used in some areas for tsetse fly population suppression. Now, tsetse flies, if you didn't know, are very important for transmission, or they are major vectors for transmission of African sleeping sickness. So there is a need for reducing the population of these flies. Now, there are other technologies which have since been used. One of them is called using cytoplasmic incompatibility, or also known as IIT. This is for population suppression. So as Professor Paul mentioned in his talk, you can use this technology to either distort sex ratios. So you can make only male mosquitoes or male insects, or you can use detrimental genes to reduce the population of certain mosquitoes. You can also use some technologies for population replacement. And by what I mean by that is you can replace a population of insects or mosquitoes, in this case, in a region with mosquitoes which are incompetent to transmit a disease. So you effectively reduce the transmission potential of those mosquitoes. Now, one technology really worth mentioning is known as Welbachia. Welbachia is a intracellular bacteria, and this bacteria is really transmitted maternally, so it's maternally inherited. So it is it produces a natural drive, so it can be driven into a population when you infect a mosquito with this Welbachia. This bacteria is naturally present in several insects. It's not present in some of the species of mosquitoes which are relevant for transmission of diseases like dengue or Zika. So in regulatory terms, it's not a gene drive. In regulatory terms, it's a biocontrol agent because you're using a bacteria, which is a biological agent, to reduce reduce the impact of these dengue or Zika viruses. And because it is a natural drive, it can also be used theoretically as a synthetic gene to drive a synthetic gene construct, although it has not really been successfully shown that it can do it. So this figure really shows how Welbachia is working, or Welbachia as a strategy can work. So these green mosquitoes, you can see, these are Welbachia infected mosquitoes. So if you use Welbachia infected females and they mate with normal wild type males, you produce a progeny which is a Welbachia containing offsprings. Now, these mosquitoes, which are infected with Welbachia, there is a phenomena which leads to its blocking of pathogens. So these mosquitoes are unable to transmit viruses like dengue or Zika or chikungunya. So these can be really used as a population replacement. So when you release this mosquito, it can, because it is maternally inherited, it can be driven into a population, and that population is unable to transmit viruses. On the other hand, if the males, you can see here in green, if they are released with Welbachia infection and they mate with wild type females, the offsprings are non-viable, which means you can use these for suppressing the population. So Welbachia has been used in various cases for either population suppression or population replacement. And it has been implemented in a lot of places internationally, as well as in very local regions. There is a lot of considerations that need to be made or have been made during its implementation. The important thing is the socio-political context and community engagement. Professor Paul already mentioned this, but it's very important for an early engagement with public to do any kind of releases of mosquitoes in that region. Another consideration that has been very important is what kind of technology we are using. So in case of Welbachia, it can be used to either suppress the population, or you can use it for replacing the population. with these female mosquitoes, which are unable to transmit the disease. So in case of suppression, you really need a large-scale facility to make these mosquitoes, and you need to do a repeated deployment to have an effect to suppress the population. In case of replacement, because it is maternally transmitted, it can be relatively self-sustaining. So if you release few times mosquitoes which are in female mosquitoes which are infected with Welbachia, it remains in that population for a relatively long time. But for all this, we need to understand what is the basic population of mosquito we are trying to replace or suppress. So, and that really depends, it's region specific. So you want to understand what is the mosquito population, what is the gene flow? So where are these mosquitoes coming from in that region? Where are the mosquitoes going from that region? So you don't want to replace a population and then find out those mosquitoes are being replaced by something else, some other mosquitoes coming into that region. So there is a lot of modeling that goes behind the scenes as well. And this all is underpinned by risk analysis. And this is based on a lot of lab experiments and field trials. And things we need to check are like other infections. So we may release mosquitoes which are resistant to transmitting dengue or Zika, but are they able to, but does that change their infection status for other viruses or bacteria? We need to check that in the lab. Is there a horizontal transfer, which means are the Welbachia can be transferred to other insects or other species of mosquitoes? Is there an escape? So we need to look at, because viruses can mutate, can the virus come, get around to the fact that Welbachia is blocking it, and there is a resistance developing in viruses. So there has to be this continuous monitoring for Welbachia established that has to be done over time. And the most important thing we need to be able to track is the effect on disease reduction, because that's the ultimate goal. You want to be able to monitor the disease status in that population. And that can be done using RCTs, so randomized controlled trials. Now, there has been a lot of, now this Welbachia technology works well, and it has been trialed in various areas, but scientists are always looking for newer ways to block infections and to reduce population. And obviously, biotechnology has made a lot of progress in last decades. And with so in mosquito area, there has been identification of transposons like piggyback. These are the genetic elements which can introduce new genes into a mosquito genome. There's always already naturally occurring selfish genetic elements. As Professor Paul mentioned, there are some genetic elements which are naturally present, which can drive a specific gene into a genome. There has been a lot of advances in identifying promoters which can drive a certain gene. So this can be tissue specific or developmental. So it can be very specific. There is the genomes of a lot of these vector mosquitoes are known because of advances in genome sequencing technologies and also use of fluorescent markers. Now these fluorescent markers can be used in lab or in sort of field trials to track genetically modified mosquitoes as compared to wild mosquitoes. So you can do that comparison. More recently, there has been a lot of targeted approach for gene modification. So you can do a precise genome engineering to either modify a single basis or single nucleotides or a small part of the gene. And this can be used using homing endonuclease genes, zinc finger nucleases, and something known as talons. I won't go into detail about this, but what I want to bring forward is the point that these technologies do exist and they have been trialed in mosquitoes to modify their genome. Now, one notable point or a technology that's worth mentioning is called Riddle technology, which is developed by a company called Oxitect. And Riddle stands for Release of Insects Carrying a Dominant Lethal Gene. And these mosquitoes, these Oxitect mosquitoes, are called OX513A. This is a name they have given. What this is a specific gene target, which really causes late larval death. So it's very specific to, so when these mosquitoes are released, these are male mosquitoes carrying this gene. When they mate with wild type female, their progeny dies at a larval stage. So this can reduce the population in the wild. Now, interesting thing is it's self-limiting. So they use tetracycline to breed these mosquitoes, but when it's released, because there is no tetracycline in the wild, these mosquitoes die off over time. So once you stop releasing these mosquitoes over a few generations, these mosquitoes are not present in the environment. So it's self-limiting. Now, there have been some releases for field trials. There was in early 2000s, there was a release done in Cayman Island, where they saw about 80% reduction in mosquito numbers over 23 weeks. There have been some field test sites in Brazil, and it was really considered as a response to Zika outbreak. So Zika virus, if you remember, caused a huge outbreak in 2015, 2016, which led to infants being born with microcephaly. So at that time, this technology was really considered, although not used. Now, if you have been following some news in the field, you will probably know that there is a first US field test site in Florida that will be trial beginning in next few weeks. So this is the Oxitect technology, second generation technology. So they will be using what they call as friendly mosquitoes, which use a technology where it causes death in females. So we'll see how these results turn out. So it's worth keeping an eye on it. Now, Professor Paul already mentioned how CRISPR-Cas9 works. The real beauty of CRISPR-Cas9 technology is the ease with which we can modify the genomes of mosquitoes with this. And this technology has been used now to replace a population. So this paper obviously mentioned before from California, which actually used this technology to make mosquitoes resistant to Anopheles, which is a malaria vector. So these all experiments have been done in the lab, and they showed that they can really replace a population in a large cage with these mosquitoes. They have also used this technology to suppress the population, to reduce the population of mosquitoes, and that has also been successful in the lab experiments as well as some large cage trials. Now, there are some technical concerns which were mentioned before as well. One of the main concern is, can you contain a gene drive? So if you release these mosquitoes in the wild, which are genome engineered with a gene drive, can you actually stop this drive of this gene into a wild population? So there is a lot of technology, a lot of research that has gone into this with this technology called split chain drive or a daisy chain drive, where different components of this gene drive are at different loci in the genome. So which can really show that there can be a containment of this gene drive over a few generations. The other part is resistance to gene drive, and this was also mentioned before, which one of the papers showed that if the gene drive, in a caged experiment, it showed that the gene drive is lost after a few generations, but it was later found out that it's target dependent. So you can change the target and you can sustain the gene drive, at least in cage trials. All of these experiments are done in sort of a large cage to check how the gene drive is progressing. So what are the considerations? What can we learn from what has been done so far? Well, the first is, and most important, is the public engagement. I told you the story about DDT, of course. Public perception can really put a stop on any new technology. So early public engagement and also throughout public engagement. And this can be done with social media as well as town hall, whatever medium can suit this particular region. The another thing to follow up is mosquito fitness compared to wild. Some initial experiments were done and some field releases that were done by Oxitect found that the mosquito was not as fit as the wild mosquito, which reduces its capacity to drive this gene into a population. Costs are obviously play a major role. There are initial costs of equipment to produce the large scale, large breeding facilities, but also there are long-term costs because these are not short-term projects. These are long-term projects, and you want to be able to monitor the gene drive trials over time. Local capability development is very important, and that also feeds into this long-term sustainability of these projects, because you don't want a scenario where an international body comes in, does the field trial, and leaves, leaving the local authorities to deal with the future, whatever the future may hold. So these are some of the considerations we can learn from previous experiments, and I just want to mention, as I mentioned in the beginning, my focus has been on mosquito-borne disease, but obviously gene drive can be used in other systems to either reduce invasive population, as Professor Thomas has already mentioned, for conservation, or as well as de-extinction. So I will stop here and take any questions you may have.
Thank you for that brilliant presentation, Dr. Prasad. I can see that there's been quite some progression in how gene drive research came about. And thank you for providing us with an overview of that, of the current state of gene drive and the knowledge and experience of similar technologies that can inform it. To ensure that it proceeds safely and responsibly. At this point, I would like to open up to the question and answer session and invite our audience to use our question and answer box to send in your question if you haven't yet done so. However, we have received some questions and at this point, maybe let's start with a question on regulation that we can see is coming up quite a bit. So someone is asking that in several countries, genome edited organisms that do not contain a transgene are not considered GMOs, hence are not regulated as such. And I would like to know that do synthetic gene drives involve transgene in the final product?
I'm happy to have a crack at that. Very much so. So at least, for example, the two drive mechanisms that I described, which involve CRISPR. CRISPR is a foreign gene that wouldn't normally be present within the genome, and to insert it requires genome modification. So they would qualify as indeed GMOs, at least in the Australian context. I'm guessing probably in most jurisdictions as well. Prasad, have you got any further comment on that?
Well, I'll just add that although there has been a lot of effort, mainly by scientists, to put together a sort of a framework where we can work in this gene drive technologies, a lot of countries are still not up to date with their regulations. So I think there has to be a concerted effort from either a unified international body or with all the scientists coming together. And that's somewhat happening. It's still lagging behind.
One other quick comment is, at least in the Australian context, the Office of Gene Technology Regulator recently amended the regulations to specifically include gene drive organisms under the legislation. So now those of us working in the area we know exactly what we need in terms of to perform the experiments, even if they're done in a safe sort of split drive manner that was described. We need a license to do gene drive development in Australia, which was not formerly the case.
Thank you for those responses from our speakers. So there's another question for Paul here, where someone is asking that, was a dramatic increase in homing rate compared to the naturally occurring frequency due to some genetic design? And can you expound on how that was done? Thank you.
Okay, so in terms of the homing gene drive, which is, I think, is what this question is referring to, they have been developed as proof of concept in Anopheles, Aedes, to some degree Drosophila, so all insect species, and yeast as well. And the bottom line is that it works very efficiently in Anopheles and yeast, less efficiently in the other insects. But we really don't know in a broader sense in other species where we might want to deploy the technology, for example, mammals, how well it's going to work. And without getting too technical, probably what it boils down to is the level and timing of the CRISPR machinery at the time in germline development, so sperm and egg development, such that you maximize the chance of the gene drive being copied over onto the other chromosome. And so far, at least in rodents, mice specifically, it's not working very efficiently. That doesn't mean that it will never work efficiently. It might mean that we just need to do a whole lot more experiments to get it working. The jury's still out. Personally, I think it might be difficult to get it to work efficiently enough to make it a viable option to actually think about using in the field, but I would be very happy to be wrong about that.
Professor Thomas, why do you think gene drive is so much less efficient in mammals than in the mosquitoes?
We don't have the answer yet as to whether it's a biological difference or whether we just haven't managed to do the experiment in the most optimal way. So as I mentioned before, each of these experiments take 1 or 2 years to get going. So there's not a lot of data yet. The simple experiments have been done. The most straightforward way of doing the experiment has been done by us and by others. And in essence, the rates of homing are not very impressive. It has worked to some degree, but not super impressive. So does that mean that we'll never get it higher? Or does it mean that we have to do more experiments to try and really hone in on what we think might be the best time to express that CRISPR machinery? The answer is as yet unclear.
Thank you for that. So quite a few individuals are wondering about the application of gene drives in plant improvements, like say crop improvements or disease control, a likely use in multiploid plants maybe. So I think the question that a lot of people want to know is, can gene drive be applied to plants?
Not really my
I'm staying
area.
silent, Prasad.
I'm not really sure, actually. There has been some work in looking at plants. I know there is some research that's happening on weeds, so for invasive weed plants to suppress the weeds, but I'm sure extrapolating that it may be able to use it in sort of other plants, but yeah, that I'm not sure about what research has been done with that.
Yeah, I've similar thoughts, I guess. I haven't seen anything in plants. Potentially it could be developed in any sexually reproducing species, potentially. I don't know much about wheat, but having 6 copies of the chromosome to me seems quite a difficult challenge to get that homing reaction to occur across that many chromosomes. So
that's a good point.
You might need to be pretty brave to take on that experiment. But hey, look, I just haven't seen any data on it, and I'm not a plant person, so I'm happy to be, corrected on that.
Right. Okay, thank you for that. So to both of our speakers, in your opinion, what are the prospects and efficiency levels for the application of this gene drive technology?
I think it will really depend on the what target animal we are using. So in case of insects, the studies show that it will be really effective. But I think unless we do those experiments in some of the other animals, it's very hard to say. I'm not sure what Professor Paul has to say.
Yeah, I think that's right. It's going to be very species dependent, seems to be the picture that's emerging. And I think it's going to be important not to put all our eggs in one basket in terms of, you know, a particular strategy, pinning our hopes on that. I think it's going to be better to try a number of different approaches. I talked about two, and Prasad talked about some of the other approaches as well in insects, some of which are insects or species, you know, specific. But we'll need to hedge our bets a little bit because it's not clear yet as to which strategies are going to work most effectively in each individual species.
And I think it's important to also understand underlying biology and the sort of behavior of each species, and that will play a huge role in using different strategies. So yeah, you're right.
Yeah, and I guess the other thing is that, you know, if we get it working in, let's say we get it working in mice, would be an example of a mammal context for, let's say it's a wire, you know, driving wire or a homing drive. That opens up the question of what about other invasive species that are mammals? You know, cats spring to mind because there's a lot of interest in, you know, feral cat eradication in Australia. We also need to consider not only the biology of the species, but the generation time, litter sizes, and all those, you know, social structures. So there are a lot of things that we need to model very carefully before we can be convinced, I think, as to how broad we would need, you know, what's the real potential of the technology. So it's not even if we had a perfectly operating drive, if it takes, you know, 300 years to cause a small population to be eradicated because of the nature of the species, then you'd have to question whether that was the way to go.
Thank you for those insights. I have another question that says, in rare cases, can sterile technology-based gene drives lead to species extinction? And if yes, how can this be mitigated?
I'm happy to have a crack at that one. Prasad showed an example of, I think it was the Daisy drive system, was that right, Prasad?
Yep.
Yeah. So I think just as important as having a drive system that works is having a safety switch incorporated into it that would limit its either number of generations or somehow fixes it on the target species and the target population that you want to address. I didn't talk about it at all, but it's going to be a really big challenge, I think, as much as getting the drive working, is to come up with the safety switch that would enable us to deploy it safely within a target population, because, you know, we do not want to eradicate every mouse in the world. You know, that's not the aim of the exercise, of course. And it's, in terms of conservation, the people in the area are very conscious of not wanting the drive to get back into the native range where the mice or whatever the species is part of the ecosystem and should be there. So those safety switches, again, it's pretty early days for those as well, but they're very important in this field of research.
Yeah, absolutely. And I think there is a question in that QA box is, can this lead to extinction of mosquitoes? And theoretically, it can lead to extinction of specific species. So all these gene drives, that the focus is on specific species of mosquitoes or species of animals. So it's not, it doesn't mean that all mosquitoes will be extinct. It's that species that we are trying to remove from a region. So again, some mosquitoes are invasive to certain areas, and the idea would be to remove those mosquitoes. So there has to be that stop gate at, you know, somehow we should be able to contain that gene drive, and that's the safety switches that Professor Thomas talked about here.
Thank you. I'm sure it's quite reassuring to hear that gene drive can be very, very specific. That is wonderful. So we do have some basic questions coming in as well. Someone is saying that thank you so much for the enlightening talk, but gene drive is still new to me, and I would like to know, one, can gene drive be applied in asexual organisms? Two, can we apply it to any genes, or does it have to be genes involved in the reproductive mechanism? And three, can the technology work in microbes like fungus and bacteria?
The homing drive, as I described it, which is a lot of what the discussion's about, is only can only be used in sexually reproducing organisms. So bacteria would be out, would be my understanding. Sorry, what were the other ones? I can't see that question. Maybe Prasad, you can chip in.
Yes.
Can we apply it to any genes, or does it have to be involved in the reproductive mechanism?
Doesn't have to be reproductive genes, but they're a good target for population suppression, which is where my interests lie. But Prasad may have other targets.
Yeah, the other targets in mosquitoes, especially, we can introduce some of the antiviral or anti-malarial genes, and that can be overexpressed to, like antibodies, we can overexpress, and that can be driven into a population to not suppress the mosquito population, but replace them with the population of mosquitoes which can block this transmission. So those are different kinds. Yeah.
Okay, thank you for that. So you also mentioned that there are gene drives that occur naturally. Is it possible for you to cite some examples?
One that's close to my heart is the T complex in mice. So it's a naturally occurring drive. It's really just a rearrangement of one of the chromosomes that enables that rearranged chromosome to be transmitted even up to 95% of offspring. So very effective way of biasing its own transmission. So there has been discussion around, if you like, piggybacking onto that naturally occurring drive, some kind of, well, the work that's most discussed is a sex reversing, so a male only type offspring drive, which would then cause all the population to become male, and similar as I described previously, like a Y drive, driving Y X shredder approach, and cause the population to crash. That's one that springs to mind. I know there's many sex disorder and transmission distortion loci in insects, but I'm certainly not expert on those. So it's very fortunate that I'm sitting virtually alongside an insect expert.
Okay, so also one of our attendees would like to know if there's been any gene drive research done in invasive fish and amphibian species.
Nothing published in fish as far as I'm aware.
Yeah, I'm not
There's
sure.
Interesting carp. Yeah, carp control and gene drives has been discussed, but I think I have heard that there's research going in, or at least funding going into zebrafish as a model system of a genetic model system in fish that could be used as a a testing ground to develop gene drive technology, perhaps for carp. But I've not seen anything published in that area.
I know there is a not gene drive, but genome modification has been done in salmon, if I remember correctly, and that's been marketed in US currently. So not gene drive, but gene modifications.
Yeah, sounds like we still have plenty of opportunity for research for different organisms, yes? Okay, so we also have a question. So Crispr or Cas9, no doubt is impressive technology. However, the Crispr Cas9 system also has certain drawbacks. And scientists are debating on whether to regulate genome-edited products or not. So how can we think about using CRISPR with this gene drive? Doesn't gene drive also have concerns?
Yeah, I mean, I think CRISPR-Cas9, yes, it does have certain drawbacks. All of the strategies that I discussed and some of Prasad's used CRISPR-Cas9. The one that's commonly cited is that CRISPR will cut sometimes in the wrong place. So that's called an off-target cut. And that is important to consider, probably more in the context of CRISPR therapy, where you don't want to, for example, create a mutation in an oncogene to create, another disease when you're trying to cure a different disease. I see that less as a concern in gene drive organisms. They're already GMOs, and if they happen to carry a second off-target cut that wasn't affecting the phenotype, then that wouldn't be a terrible, necessarily a game changer. So yes, except
probably, sorry to jump in. Except probably if there is a fitness cost, isn't it?
Yeah.
Okay.
If it doesn't have a detriment, though, I don't think it's necessarily going to be a problem. And to some degree, they can be predicted in advance based on the sequence of the guide.
Yeah. Okay, thank you. So we also have a question. I think it's from someone in Nigeria saying that their question is the issue that they have with Nigerian mosquitoes that resist most of the insecticides applied to them. The insecticide does not kill them completely, but rather reduce the activity for some minutes. Do you think gene drive application will serve better? Have you ever found out about that issue?
So this is the whole basis of looking at novel technology. So yes, until now, really, insecticide has been used successfully in some cases, but that's where you're now finding mosquitoes becoming resistant to insecticides, and that's what I guess we are seeing all over the world now. The insecticide resistance is increasing. So gene drive can or may provide maybe a good strategy to deal with that. But it also comes down to whether that's something that, because it is a new technology, obviously there has to be a lot of regulatory hurdles and public engagement that has to be done before we can consider that. But theoretically, yes, it should be able to work.
All right, thank you for that. So this is a question for Paul. Dr. Prasad made a great presentation on how the fight and tools against vector-borne diseases evolved throughout time and how gene drive research fits into that. And so the question is, Professor Thomas, which precedents do you believe were also relevant for gene drive for conservation purposes?
I think it's pretty early days in terms of for conservation purposes. So we've had examples of biocontrol here in Australia that involve, for example, we have a rabbit problem, and effective biocontrol has been used in the form of a virus that's able to spread horizontally between individuals to cause the population to become sick and therefore decline. So initially myxomatosis and then calicivirus. They've proved to be reasonably successful biocontrols in Australia. So there's, if you like, there is a precedent for biocontrol in the broadest sense, but what is definitely new about gene drives is that we could potentially use the natural mating processes that occur to transmit a genetic modification to cause population suppression. And that's quite new. Why do we want to go down that path? Well, again, I'll refer to something that's happening at the moment, and that's mouse plagues. We've had mouse plagues in Australia for 150 years. In fact, ever since you know, the early settlers came out, they brought rodents with them. So at the moment we have baiting as a strategy, which has been around for a long time, and it's a band-aid solution. So there's a lot of interest in new technologies that could have a landscape scale effect in terms of controlling rodent populations that do enormous damage in agricultural and conservation contexts, and to some degree human health as well, certainly human mental health. So there's not a precedent for genetic biocontrol, but there's a precedent for biocontrol in Australia. And I think there's a lot of interest in looking for new solutions. And what's going to be really interesting to see is what the social acceptance of a new form of control technology is from the stakeholders and the general public in this space. So precisely because there's not a lot of precedent for this specific type of technology. I think what's happening in the context of malaria is going to be really interesting as well, because in a sense, that's a very urgent need for the technology. And if that proves to be successful, I think, you know, that could provide a useful example of what genetic biocontrol can do. So very early days, and there's really not much precedent yet.
Thank you for that, Professor Thomas. Unfortunately, we don't have much time left, but I'm sure we've addressed some key aspects of gene drive approaches during the Q&A and our presentations today. So thank you all for this fruitful discussion. It was really interesting to see how gene drives are a genetic tool that can be used for population modification or suppression, and how useful it is likely to be in malaria control in the near future. We hope, fingers crossed, and also to the considerations for gene drive technologies, societal acceptance, risk assessment, and also to understand how we'll need to do some things like computer modeling and regulatory approval to get it done. I also have some takeaways from Dr. Prasad's presentation, that vector-borne disease control needs novel strategies, including vector control using gene drive. Population suppression and population replacement are two major strategies that we can use to do that. And also that historically, any new strategy requires various considerations targeted towards regions, including public engagement, costs, and sustainability. So I think that we've had a very useful and fruitful webinar today. And at this point, I would like to finalize our event by inviting Dr. Rodora Romero Aldemita from ISAAA to say a few words.
Hello once again. Good morning, afternoon, everyone, to all who have stayed on. I would like to thank everyone who have responded to the pre-webinar survey. And now I would like to invite everyone to respond to the post-webinar survey. The link is now posted at the chat box, and this will help us determine if you have raised your understanding of gene drive and its applications, as well as to obtain your valuable suggestions in the improvement of our online exchange. This will also be the basis of your certificate of attendance to this webinar. So I'm also inviting everyone. So now I hope that you have seen the link to that post-webinar survey. Please respond to it so that you can have your certificate. And currently we are also preparing for the next webinar. So I'm also inviting you to our next webinar, which is entitled, Could Gene Drive Approaches Be Used to Protect Nature and Recover Biodiversity Loss? So I think EJ can share the poster now. Yes, so this is now our next webinar. So it's going to be, it's entitled, Could Gene Drive Approaches
be used to protect nature and recover biodiversity loss. So you can see the three speakers and our moderator. This will be sent out via our usual channels of the ISAAA Crop Biotech Update and all our social media outlets. And if you respond to the post-webinar survey, we'll send you the certificate and of course the link or the registration link for webinar number 2. So I now would like to close our wonderful webinar. So on behalf of ISAAA, the host of this webinar, the Outreach Network for Gene Drive Research, and the ISAAA Biotechnology Information Centers, we would like to thank the panelists, Dr. Paul Thomas and Dr. Prasad Paradkar, for their comprehensive presentation and for the engaging conversation during the Q&A. Thank you so much. Let's give them a virtual round of applause. And we would also like to thank our moderator, Dr. Crystal Birungi Mwesinga, for her excellent facilitation of the webinar and her additional comments during the Q&A, and of course, for giving us the summary of the whole webinar in the form of key takeaways. We would also like to thank the active and excellent participation of our audience. We're very happy that, we still have to ask Dr. Paul Thomas if he's willing to share your PDF, the PDF of your PowerPoint slides. Yeah, yes, if we can share it. But of course, the video of this webinar is going to be posted at the YouTube channel of ISAAA, and we are on Facebook live streaming. So anyone who would like to see it again, to learn more or to get more in depth in the discussion, please look over at the YouTube channel of ISAAA and also the Facebook. And so thank you very much, participants. We have reached 189 today. And at the close of this webinar, I can say that we have contributed to the understanding of the gene drive technology, which we hope could promote a productive and balanced conversation on the benefits of gene drive applications, which could hopefully move enabling policies in our region. We know and we have seen that a lot of questions were raised on the science, so very few people know what gene drive is, and we found out also in the pre-webinar survey. So ISAAA supports the understanding and application of gene drive technology as we see its many potential contributions to food security, environmental sustainability, and in the improvements of human health. As such, as ISAAA's facilitation of information and education on biotechnology extends to other sciences such as gene drive, we help prepare the general public and various stakeholders in the acceptance of the technology for the need of the global community. Thus, I would like everyone to subscribe to our newsletters. So we have these 3 ways of getting more information on biotechnology that now includes gene drive. So we have the Crop Biotech Update. We have 25,000 subscribers, and this is published from the ISAAA Southeast Asia Center. And then we have Drumbeat, which covers Africa and published from the ISAAA Afri Center, and the Petri Dish from Malaysia Biotech Information Center. So all of these are online, and you can subscribe to this all free. The newsletter comes to your inbox. So with that, I'd like to thank you all once again. Let us continue to be educated on biotechnology, especially gene drive, as we see you on June 24. Thank you very much. And back to you, Maha.
Thank you very much. And I think, Ola, you covered everything. ISAAA is not just about crop biotechnology. We are very happy to move into other very important gene technologies, and gene drive is certainly one of the technologies. as I said in my opening, it is complex. So we invite scientists and other stakeholders like you to submit articles as well. And ISAAA will also be happy to do capacity building programs, especially in communication. Like one of my colleagues here, Dr. Margaret Karambu from Africa, she said that the tone has to be set right for the public and other stakeholders. So we'll be very happy to support that initiatives. So I don't want to be taking your time. Thank you so much again.
Okay. Bye-bye for now.
Yes, bye-bye and see you in the next seminar.